So as an overview of the structure of DNA, it's first important to note that DNA is a double helix. So it is a double helix, which essentially means that we have two strands of DNA. So it could be strand 1, and this could be strand 2, and they're wound together. And we can see this right here. So we have two strands of DNA wound together, forming a double helix structure. We have three different forms in which DNA can occur: the A form, the B form, and the Z form. And they all differ slightly in their conformation, and we'll go over how they differ in their conformation. So the A form of the DNA, if we quickly look at it, it's actually the shortest, whereas the Z form is the longest. And there's reasoning behind this. But within biology and biochemistry, the most prominent form of DNA that we see in depictions and illustrations in our textbooks and notes is that B form. This is what we're used to. So what's the difference between the A form and the B form? Well, we can note that they're both anti. Anti refers to the bond, the orientation of the bond that forms between the sugar and the nitrogenous base. And we'll go over that in a second, but remember, in DNA, the sugar is deoxyribose, and the nitrogenous base could be a pyrimidine or a purine. So they could be adenine, guanine, cytosine, or thymine. So when we talk about the bond that forms between the nitrogenous base and the deoxyribose sugar, we say whether it's anti or whether it's syn. And in the A and B form, it's anti. Now, the A and B form are both right-handed, which means the turns happening in the A and B form are right-handed. And so when we talk about the sugar, the deoxyribose sugar, they differ in their positioning. So here we can see that in the A form, we have a C3 endo positioning, and in the B form, we have a C2 endo positioning. And that's what actually results in the difference in the length of these both forms. So the difference in length of these two is a result of these characteristics. Now, the Z form is a bit more complicated because it includes the anti and syn form and the C2 and the C3 endo. So the anti form can exist in the pyrimidines, the syn form exists in the purines, pyrimidines are C2 and the purines are C3, and it's a right-handed turn. So the A and B form are actually quite similar. They only differ in their C2 and C3 endo positioning, whereas the Z form, it's right-handed. The A and B form, oh, pardon me, there's a mistake right over here. This should actually say left-handed. So the A and B are right-handed, and the Z form is left-handed. And now let's go over what the anti and syn means. So remember I said that the bond between that deoxyribose sugar and the nitrogenous base depicts whether we are anti or syn. Now, this bond that forms right over here, so this is our deoxyribose sugar, and this is just an example. So in this example, we have used adenine, which is a purine, and this bond is called the N-C-glycosidic bond, and it's free to rotate. And since it's free to rotate, it can exist in two conformations: the anti, in which it kind of points away from the sugar, and the syn, where it kind of points inwards towards the sugar. So when we say that the A and B form of DNA are anti, we essentially mean that this is the orientation that's occurring, where the nitrogenous base points away from that sugar, because this NC bond is a glycosidic bond which is free to rotate. Whereas in the Z form, when we see that syn conformation in the purines, it's as a result of the nitrogenous base pointing inwards towards the sugar. Now, when we talk about C2 and C3 endo, that also refers to the sugar. So here we have a chair conformation of our sugars, and we can note that in the C2 endo form, the phosphate groups on C5 and C3 are far apart from one another, 7 angstroms, whereas in the C3 endo position, the phosphates are a bit closer together. They're at 5.9 angstroms. When those phosphates are closer together, it's called the C3 endo, and it is found in the A conformation. So whenever the phosphates are closer together, it actually results in the closer, it results in the shorter length of the DNA double helix, and as a result, we get the A conformation. So the shorter length is the result of the phosphates being close together. Whereas when the phosphates are further apart, we get that longer B form. So C2 endo versus C3 endo. In C3 endo, the phosphates are closer together. In the C2 endo, the phosphates are further apart. And C2 endo refers to the B conformation, and C3 refers to the A conformation. So now we just get a little bit of sense of the different forms of DNA. One interesting fact to make note of is that if we were to have a strand of RNA to bind, to form a helix with a strand of DNA, so let's say we have this RNA, and then we have another strand, and we can say that this is the DNA. So this is DNA, this is RNA. The conformation that this RNA DNA helix, it will actually exist in the A form. So it will be an anti-right-handed C3 endo conformation. And that's just an interesting fact. So that's essentially an overview of the structure of DNA.